Thank you very much, Elaine, and thank you for hosting this event on behalf of Oxford's Biomedical Research Centre, and a special thanks to the Biomedical Research Centre for providing the drinks and nibbles. So I wanted to first of all start just by introducing a little bit about vaccinology in Oxford and some of the background to why the study that we're going to talk about took place. And first of all, just to put in context the work that was done on this study is more widely in Oxford in the Jenner Institute, which is a fantastic collaboration of different institutes around Oxford which are all involved in vaccinology. Just next to us here in the green building are the Jenner Laboratories, which Adrian Hill leads, working on various different vaccines that are in development, including a novel influenza vaccine, which is obviously very relevant for the discussions today. Over on the John Radcliffe site is work on HIV vaccines. Down on South Parks Road is work on hepatitis C vaccines. And we also work very closely through the Jenner Institute with the Institute of Animal Health in Compton, where again there is work going on in influenza vaccines, and as you'll hear, that's very relevant to animal health as well as to human health. And we also have here a biomanufacturing facility which is used to make some of the vaccines that are being tested here in Oxford to try and improve the health of both adults and children around the world. But we're talking today specifically about a trial that was undertaken at the Oxford Vaccine Centre, which you see here, at least that's where it was led from. We're going to talk about the various pillars that we'll put together to hold that up. I mentioned some of the different themes that there are in vaccinology in Oxford. There's my group here, which has works on mainly meningitis vaccines, pneumonia vaccines, and typhoid. We also obviously have worked with on other things, including influenza. There's a TB vaccine group, and I've mentioned HIV, flu, hepatitis C, and Adrian Hill's malaria vaccine. Some of the most important vaccines for improving health, both here and also in developing countries. So my group, which was involved in this particular study, has been involved in studying vaccines since the early 1990s, and we now conduct studies across the whole of the Thames Valley. And there's over 60 staff who work in these studies, mostly on paediatric vaccines, vaccines for children. We have also studies in developing countries in Togo, Nepal and Fiji that are ongoing at the moment, and also a laboratory program. Today, the swine flu study is over, but there's about 30 projects still running. There's about 2,000 children and adults across the Thames Valley in vaccine trials that we're involved in. As you can see, there's an array of different funders, and I think particularly important in this forum is that one of our major funders is the Oxford Biomedical Research Centre. And the reason why we're all here, and this group of individuals who work in vaccinology with me, is to improve the health of children and adults through immunisation. And I'm joined on the podium today by Professor Miller from the Health Protection Agency, who has been leading the immunisation division at the Centre for Infections at Colindale, where a lot of the work there is on surveillance for infectious diseases. It's one of the leading centres for influenza surveillance and also laboratory methods for detecting both the virus and the immune response to the virus. And Liz has been very active in studying vaccines which are part of the Department of Health's immunisation programme, interventions which every child in this country has early in their lives. The Centre for Infections, through Liz, also provide advice on immunisations to health professionals across the country and have a major role in informing national policy, particularly again through Liz's advocacy in the World Health Organization, informing global policy on immunisation. So to move to influenza, you'll all be aware of seasonal flu, which we hear about, that fills the hospitals, and I know that for Elaine this is a headache every winter with the bed crisis that it occurs. And this is the influenza virus, and you can see that the surface of it has these spikes sticking out, which are the spikes which the immune response sees and that the vaccines are developed to produce an immune response against. These seasonal flu viruses change a little bit every year, so that each year we have to develop a new flu vaccine, and here's an example of one of them, that will work against the virus that's circulating that year. And that is a major burden on health systems. There's a big cost in developing those vaccines each year. But the virus is changing a little bit. And although we have many, many cases and a high burden of mortality from it, it is a degree of illness which is sort of accepted by the population. I'm not sure whether it should be, and I think probably influenza has been under-recognised in terms of its importance in our health systems, even in wealthy countries. But really the thing that hits the headlines has been pandemic flu, which is really the story doesn't start in 1918, but it's where people are most familiar with it. And this is a ward in Camp Funston in the US in the 1918 Spanish flu pandemic with large numbers of individuals involved. And we know that during that pandemic, something like 50 million people around the world died from the virus, and that really set the scene for the fear there was with the pandemics that came subsequently. And we've had two that were well documented. There was the 1957 Asian influenza pandemic, where there were about 1 million deaths documented. And then in 1968, the Hong Kong influenza pandemic, where there were about another 1 million deaths. We think, according to the WHO anyway, that the numbers of deaths that we can document with the swine flu pandemic of last year is around about 18,000, according to the most recent figures on the WHO website today. And so clearly on that scale, this has been a very different experience, and Liz Miller will talk more about some of the epidemiology around the H1N1 swine flu virus in a moment. But really the recent vaccine history around pandemic flu starts with avian influenza in February 2003. So we know that influenza viruses are viruses which are very common in birds, particularly in crowded settings, and that's most commonly found in East and Southeast Asia. And in those settings, there are a lot of influenza viruses circulating both in wild birds and also in domestic, well not domestic, but birds that are being kept for food. We know that at the moment all of these different viruses over the last 10 to 15 years have been circulating at high levels in birds, and H5N1 was one of those, and that is what became known as the bird flu virus when in February 2003 two individuals who worked with chickens were infected by this virus and died. And since then there's been a large number of cases reported mainly from that region of the world. And really from February 2003, as the numbers of cases increased, a decision was made that we needed to prepare ourselves in case the bird flu virus became able to transmit readily amongst people and cause a pandemic. And we got involved in that as well here. This was the Oxford Mail with the large chicken and a rather small version of me next to it saying, "Help beat bird flu." And we were involved in one of the trials that was conducted to try and develop vaccines which would work well against a pandemic virus. We knew that it was a new virus, and we also knew that immune responses to new viruses are quite poor. And so a lot of work was done to try and design vaccines which would produce better immune responses and make it easier to protect the population if there was a pandemic with this devastating virus. But obviously in a pandemic, there's a lot of people to vaccinate. It's not just taking a small number in high-risk groups. I think all of us would want a vaccine if half of the people who got infected died, which is the case so far with the bird flu vaccine. And so the other thought was, could we make a vaccine where you could use lower doses so that you can actually spread it out amongst more people? And also there needed to be work to make the vaccines more quickly. There's no point in having a vaccine for pandemics that still takes 6 to 8 months to make, by which time the pandemic's over and everyone's dead. So there were new processes needed to be agreed to go from the idea of making a new vaccine to actually having something which could be given to people to potentially save lives. And that process was developed around bird flu vaccines to try to put us in a position where when the next pandemic came, we would be ready to move forwards rapidly with a vaccine that could be used to try and protect against that pandemic virus. That hasn't happened so far with the H5N1 bird flu virus, but the work that was done on mocking up a pandemic using that virus is really what led us to the point we're at today, where the swine flu pandemic, where the swine flu pandemic response could happen. Winding back again to the point of time when we knew that the two vaccines were being purchased by the Department of Health. And it is called swine flu, and as Liz pointed out, it does seem a bit odd to call it swine flu, given that although it has some pig genes and it also has human genes. And in fact, there wasn't a documented direct transmission of the virus from pigs to humans. But if you look on the internet and try and find out a little bit more about the transmission from pigs to humans, you come up with this story, which says pigs catch swine flu from humans. And there is very clear evidence that humans have infected pigs. And I was particularly struck by this story. You probably can't see it very clearly, but the farmer who got into trouble over infecting his pigs, it turns out his name was Ian Pollard. And he has more than 200 pigs in Canada, and they caught swine flu from him. So essentially what happened was the Department of Health purchased two vaccines. There's one here which was just mentioned, Pandemrix, which is, they're both killed viruses, so there's a chemical process that kills the virus before they're made into a vaccine. And this one, there's a further process which then splits the virus open. And so there's a bottle here which contains that split open virus which has been grown on eggs. And it's then mixed with this emulsion, which is an adjuvant called ASO3B. And that adjuvant contains some danger signals for the immune system to tell the immune system to switch on and make a good immune response. So when the vaccine is given, it's these two bottles are mixed together in order to then administer the vaccine to the individual who's been vaccinated, with the idea that that would produce a good immune response. And as I mentioned, there was a bird flu version of this vaccine, which we already knew produced good immune responses both in children and adults. The other vaccine is this one. It's called Kelvapan, and it is a whole virus. So it's, rather than splitting the virus open, the virus is killed and kept as a whole virus. And it's grown in a process that doesn't require any eggs. And particularly for children, where we have a very high proportion of children in the population in the younger age group who are egg allergic, this sort of vaccine may turn out to be very important in a pandemic situation in the future. And this one has no adjuvant in it, so it doesn't have the additional danger signals that might improve immune responses. So the Department of Health had bought these two different vaccines for use in this pandemic. There was relatively less information about this vaccine in children and adults. So, as Liz said, there was a collaborative group put together, and these are the principal investigators at each of the sites here. Matthew Snape, who's in the audience, here in Oxford, Saul Faust in Southampton, Paul Heath here, who is at St George's Vaccine Institute in London, and Adam Finn, who's at Bristol Children's Vaccine Centre, and myself in collaboration with Liz Miller. I'm sorry, Liz had gone home just before this photo was taken, so it wasn't included. So it's a collaboration of these groups that got together to run this trial around the south of England. So why do a trial? Well, I think a lot of this has already been covered by Liz, but we didn't know at that stage what sort of impact the pandemic would have. But we did know that if it had a big impact and there was a high infection rate, a high hospitalisation, or worse still, a high mortality rate, that millions of doses would be given to children. And it would be very important to know more about the two vaccines that were going to be rolled out, perhaps to choose which one was the most appropriate for young children, and also at least to be able to advise parents about what to expect. We also wanted to know how it would be best for the UK to proceed. I'm having a vote of no confidence, I think, at the back there. But we already knew from both seasonal flu vaccines and the H5N1 that although this sounded very new with these swine flu vaccines, we had a lot of experience already of using flu vaccines. So although, as Liz said, we didn't have much information about these particular vaccines, there's a long history behind them which helped us. But why do this study in children? Because children are different. We've already heard from Liz that young children make absolutely rubbish immune responses to seasonal flu vaccines. And so we really did need to find out whether these new vaccines would be better. And of course, children have often not met flu before because they just haven't had an opportunity to be exposed. And we heard with the swine flu virus that older adults have already met a similar virus before, so already had some protection. And we know that if you've met flu before, you tend to have better responses to the vaccines. And the other thing is that although the vaccine manufacturers have, as part of their licensure process, to do some studies of their vaccines, they never compare the vaccines together because it's not in their interest to do so and they don't need to for licensure. So if we wanted to really know in the same group of children which of the vaccines performed better or whether they were both acceptable, we needed to do that in the same group. And also it's terribly difficult to actually do the testing for the immune response for flu, and those tests need to be done in the same laboratory. And so really the only way that this could be done to provide the data was to do a head-to-head study. And so this is a timeline of what happened. And for those of you in the audience who are involved in clinical research, I know there are a number are, it really was a remarkable speed of activity, partly for the efforts that all this collaborative group working with Liz put together, but also because of all the agencies involved made huge efforts. Essentially there was a call from the Department of Health to fund swine flu studies here in June. And so we then started to put things together and put in a submission of this grant at the end of July. And we had, by about 3 weeks later, had provisional funding approval and then final approval by the 1st of September. At that point, we then needed to put in submissions to the Ethics Committee and there may be people from the Ethics Committee here in the audience, and also to the R&D departments in all of these centres, and also to the UK regulator, the Medicines and Healthcare Regulatory Agency. And all of these then were put in, and we had final approvals for the ethics and MHRA on the 18th of September, NHS R&D by the 21st, and then recruitment could then commence on the 26th. So you can see that this is, for all of the approvals, and the first child enrolled in the study was only 26 days. And for looking at people who are involved in putting in submissions at the moment, and it was quite a remarkable feat. And this is, I think, a testament to what could be done when the necessary agencies are told by senior officials in government that they need to get on and do things properly. Unfortunately, that isn't our experience since this study with any clinical work that we've done in trials. So then the recruitment started, and we, across those sites in the south, within about a week, about 500 children had been enrolled. And by the end of about a month of recruitment, nearly 1,000 children had been enrolled. So, in fact, there were 949 children in total who took part. And despite here, the Oxford Mail said, Oxford swine flu trial will use babies. So that was one of the local headlines. And despite that, we were very pleased that many families took part here in Oxford. And these children were between the age of 6 months and 12 years and were randomised to receive one of the two vaccines. We did exclude a few children who wanted to take part, one who actually already had the vaccine and therefore wasn't appropriate. 4 of the children refused to have a blood test, which was required as one of the initial procedures in the study, and one of them was unwell on the first day of the visit. But there weren't any others who were excluded at the start for any of the other reasons, although we did have various criteria that we would have used if they'd had. And of course this is the alternative, is just to wear a mask, which is probably nicer than being in a study. So the design of the trial was that on the first day all of the children had a dose of vaccine and they had a blood test to measure their antibodies in case they'd already met the virus, because as you heard there'd already been a first wave of flu, so they might have met the virus already. And then 3 weeks later they got a second dose of vaccine, and then 3 weeks after that they had a second blood test. So there was just 2 blood tests at the beginning and at the end of the study, 2 doses of vaccine, and we kept detailed information in diary cards and through phone calls after each of the doses of vaccine to try and get a picture of how children tolerated these vaccines. And the diary cards and all of the information was very rapidly collated and sent to Liz's team at Colindale to then start analysis as the study proceeded. And the blood samples which were taken were also batched and sent to Colindale to the flu laboratory for analysis. There was a lot of coverage. I'm not going to go into too much more detail about the practicalities of the actual study itself, because unfortunately there isn't an internet connection here to show you any of that. But we were very well supported by the BBC's medical correspondent who came and covered the initial immunisations here in Oxford and wrote a number of stories about it. And also the parents who were involved were extremely helpful in talking to the media and explaining why they wanted to take part. And here you can see one of the volunteers, Nicolette Wolfe, who's 17-month. And here you can see one of the volunteers, Nicolette Wolfe, whose 17-month-old Reuben was immunised. And she is pregnant, and she said that she would be one of the first, once it was available for pregnant women, to be vaccinated, because she was so aware of the data that there were around the risk to pregnant women. So it was the great support from the media and also from parents in interacting with them. And I think the feedback we've had is that most of the families who took part had a very positive experience of being involved. And at this point I was going to show you some of the media stories, but unfortunately the internet connection doesn't work. So there are two different ways that we can measure immune responses to flu vaccines that are routinely used. And you don't really need to know the names of these tests, but I just show this picture to say there are two different methods that we use. And so they're different measures of how well these children had responded to the vaccines. And we'll call one MN and the other we'll call HAI. And this first graph is showing just the proportion of children who made a good response to the two different vaccines using one of the assays, MN. And so this number just means it's the level at which we believe that protection might occur. So the first thing to see is that whether it was the adjuvanted vaccine or the whole virion vaccine, before they had their first dose, only a very small number of children here in the study in Oxford actually met the virus. So Liz had shown very high rates of infection in children in London, over 50%. But here, these are just a few percent of children overall in the study across the south of England, most of whom were outside of London, had actually been infected. And so they had their first dose of vaccine and then another dose 3 weeks later. And then these are their blood test results after the second dose. And you can see that the adjuvanted vaccine overall in both the under 3s and the over 3s has got higher levels of protection in the population than the whole virion. But they're not that different. They're both quite acceptable for flu vaccines in producing protection in those children. And then we can look at the same thing using the other test, the HAI test, and a different cut-off, but another way of seeing whether children are protected. And this is perhaps particularly important because most of the licensure of flu vaccines is based on this test rather than the first one. Again, using this test, a very small proportion who had any evidence of infection before their first dose of vaccine, and a very similar picture after the 2 doses, with the adjuvanted vaccine showing higher levels of protection than the whole virion. So this is what these two graphs have been, is showing the proportions of children protected, and it gives us great reassurance that both vaccines would have been good to use in this pandemic had it been resulting in a very high mortality in children. It would have been a very good argument for rushing for everyone to get their children vaccinated with either of these vaccines. But there was a greater chance of being protected with the adjuvanted vaccine. And we can look at that in a bit more detail by looking at the absolute level of antibody in children of different ages. And so this isn't the proportions who are protected, but the actual level of antibody. And there's such a dramatic difference here now when you look compared to the adjuvanted vaccine compared with the whole virion vaccine. In the under 3s, dramatically different levels of antibody that you can measure, levels of protection in the blood. And the other interesting thing is, remember we talked earlier on about how young children make rubbish immune responses to flu vaccines. And this is quite astonishing that in fact the best responses in this study were in children under 3. And this is something which I think needs further exploration because that hasn't really been seen before with any other studies that we're aware of. So I think this shows that certainly this vaccine was very much more immunogenic, better able to make immune responses than the whole virion vaccine. So another very important component of the studies was what were the reactions like? Did children really suffer from problems with local or systemic reactions? And the way I've drawn these graphs is what were the reactions after either the first dose or the second dose? And if we first of all look at the adjuvanted vaccine, the pinky colour is after the first dose and the yellow after the second dose. And you can see that generally the trend for whether it's having pain or redness, there seemed to be more of that, of the more severe local reactions after the second dose of the vaccine, and that was one of the things that led to the Department of Health's recommendation, as you heard, to use one dose. And the other thing to notice, if we now look at the whole virion vaccine, is that the adjuvanted one seemed to cause considerably more local reaction than the whole virion vaccine, and all of these measures that we have, so either the first or the second dose compared with the first or second of the adjuvanted vaccine. And this is in 5 to 12-year-olds who are pretty easily able to tell you whether it hurts or not. So here for pain, redness and swelling. To some extent, we all expect a bit of pain from a vaccine, and I'm not sure how important this information is, although some of the individuals did complain of pain which was fairly severe. But if we now look at systemic reactions, I should say that these are all proportions of children along this axis. So if we look at the systemic reactions, the more severe reactions such as fever, you see that again the story is similar. So here loss of appetite after both the first and second dose. The adjuvanted vaccine causes more reactions. The same with feeling unwell, with headache, to some extent with nausea and vomiting and diarrhea, but not very different for those. Muscle pain was more common after the adjuvanted vaccine, as was joint pain perhaps after the second dose. But here importantly fever, which has higher rates after the adjuvanted vaccine for both the first and the second dose. So I think overall that shows in the older children that there were higher reactions again for the adjuvanted vaccine, and particularly important there with fever. So what about in the younger children? Well, it's pretty much the same picture. Parents' perception that their child had a painful arm was more, again with the adjuvanted vaccine, particularly after the second dose, as you can see here, for pain, for redness and for swelling. And in this group, who perhaps don't complain as much as the 5 to 12-year-olds, the parents were not really aware of very much reaction at all with the whole virion vaccine. They made very few complaints of that. So it's much more striking here that the adjuvanted vaccine caused more reactions. And if we now, in those young children, look at the generalised reactions, such as decreased feeding, decreased activity, being more irritable, crying, vomiting, diarrhea, it does appear, certainly for some of these ones, as if the adjuvanted vaccine has more reaction rates. But perhaps most importantly is this here, where it really stands out, that after the second dose of vaccine there were much higher fever rates with the adjuvanted vaccine. And that probably relates back to it's the febrile children who are irritable and crying more and not feeding so well. So I think that shows that the contrast between the two vaccines, that we have the adjuvanted vaccine, which tends to cause a few more reactions, but it gives a better immune response. And I think that's probably related to those danger signals that the immune system is getting to cause some inflammation and make a better immune response. But then we do suffer with that by getting these children then suffering from more reactions. So this study was just recently published in the British Medical Journal, and I think it is the first direct comparison of these two vaccines that has been done, and I think it's provided some very important data. Both vaccines did perform well in the studies, and overall, although there were more reactions with one than the other, they were pretty well tolerated, and we saw very good immune responses with both of them. But the adjuvanted vaccine was certainly the one which produced the best immune response, and in fact it was also had the lowest dose of the virus in it, which would be very important in a pandemic where you're trying to spread the doses of vaccine out amongst as many people as possible. And I think, as Liz pointed out, the observations with this study perhaps do signal the possibility to make better vaccines, particularly for young children, using some of these new adjuvants. So I think this has been a very important study, and we're really very grateful to all of the parents who took part in this study and their children, and also obviously for the large number of staff who were involved. Just here in Oxford we had to employ 80 staff in order to deliver this study over the course of those weekends that all the recruitment happened. And so we're very grateful for all those individuals, and I know some of those people are in the audience this evening. We were funded, as you heard, by the Department of Health through the National Institute for Health Research, and it was, I think, a very exciting collaboration between the Health Protection Agency and the 5 centres that were then involved in the study around the south of England. And I think we're also all very grateful here in Oxford for the support we've had from the Biomedical Research Centre, not just for the drinks and nibbles, but in making it possible to have the capacity to deliver studies like this. So thank you very much.